Welcome, everyone. It's my distinct privilege and honor today to introduce Dr. Andrew Ewald, who's joining us today as professor and director of the newly formed Department of Cell Biology at Johns Hopkins University. He also has secondary appointments in oncology and biomedical engineering. Dr. Ewald had a bachelor's degree from Haverford College of Pennsylvania in physics with a minor in biophysics, and then went on to Caltech to complete a Ph.D. in biochemistry and biophysics. He then completed a postdoctoral training with the legendary Dr. Zena Werb in epithelial biology and breast cancer at UCSF. His research has been quite influential in the field on how epithelial cells acquire the ability to metastasize, and he's developed very advanced organoid systems throughout the years to look at how distinct cell types within the microenvironment and interact with breast cancer cells, also how normal mammary glands develop, and also how the the extracellular matrix regulates metastasis. And so these efforts have extended more recently into looking at human samples and also the expansion into other cancer types. He's currently the PI on two multi-PI U01 awards, a project leader on a U54 award, and a long track record of funding, NIH, DoD, BCRF, and Metaviver. And more recently at Johns Hopkins, he's received both the Provost and the Catalyst Award from the Provost. As of this morning, he has over 15,000 citations in journals such as Nature, Nature Protocols, Cell, Developmental Cell, and others. He's the founding co-director of the Cancer Invasion and Metastasis Group at Sidney Kimmel, and training to date has included over 30 trainees, many of them MSTP students who went on to do pathology residencies. He has faculty at multiple schools in Texas and also Fred Hutch. But however, the strong mentoring record extends beyond any formal arrangement. I first met Dr. Ewald 5 years ago at Charleston at MUSC through my mentor, Mike Ostrowski, and he's been a source of wisdom and guidance since then on emails that I've sent to him with questions. And the first time I saw him speak, I was told I was in for a treat, and we all are today. Welcome, Andy Ewald, to UAB.
Thank you so much for the very generous introduction. It's a pleasure to be here. Shame we can't be together in person. I will try to start the slides here. All right. I have no financial relationships relevant to the material I'll present today. So I'll talk today about molecular and cellular mechanisms of breast cancer metastasis. And I want to first, I guess for the Department of Pathology, this is a less necessary slide, but we are very interested in metastasis because it is the major clinical problem in breast cancer. This is what drives most bad outcomes in breast cancer, as well as across solid malignancies. So as you well know, outcomes from locally confined breast cancer, so if the tumors confined to the breast, are outstanding. And even with spread to regional lymph nodes, the overwhelming majority of patients do well. However, at metastatic stages, when these tumors spread to distant vital organs, it becomes a much more dire situation for the patient. So I would love to tell you that I'm about to explain exactly how metastasis works in cellular and molecular detail. We've totally, completely figured it out. And that is not the case. I'm going to give you what we think are important pieces of the puzzle. But it is very much still a work in progress in our lab and across labs around the world. So why is that, right? This is the major driver of deaths from cancer. Cancer is the #2 killer of Americans. Why don't we totally understand metastasis? And I think there's really 4 different things that come together to make this a particularly hard problem. The first is that metastasis occurs deep inside the body. That makes it very difficult to observe, very difficult to experimentally intervene, and challenging, though not impossible, to recapitulate the necessary conditions to model the biology of metastasis. Second, there's this issue of timescales where there's important dynamics clearly occurring from seconds to decades, which makes it difficult to understand where to focus. I'll also tell you, in my experience, it's been difficult to recruit graduate students for projects that are going to take decades. So we really need to figure out, how do we study the most important parts of this problem in a way that's experimentally tractable? Third big issue here is that tumors are highly complex. Again, I'm preaching to the converted here and to those more familiar with the structure of tumors than I am. But tumors are highly complex, and that creates 2 separate problems for researchers. The first is that It's difficult to know what to focus on. There's so many differences between an advanced tumor and a normal duct. And the second distinct problem is that a lot of basic scientists really prefer simple problems, and metastasis is not a simple problem. So it has at times been difficult to recruit a critical mass of researchers to work on it. And then finally, I think bringing all these things together, the biology of metastasis is inherently importantly organized at the molecular, cell, tissue, organ, and organismal level. So this is inherently a systems biology problem. We're going to need to bring together expertise across length scales and scientific disciplines. So I was very pleased you mentioned Zena Werb. She was incredibly influential as well as a dear friend. And I didn't actually join her lab to study metastasis, as important as it may be. I joined her lab to understand how the normal mammary gland develops. So this shows a 3-week-old mammary gland and a 10-week-old mammary gland. And you can see there's a rudimentary ductal tree in the upper image on the left that expands dramatically in size and complexity over this 7-week period. And I wanted to understand how that worked. And what I knew was there was a whole series of papers at that point that had knocked out a gene or overexpressed a gene and been able to show that, hey, look, the migration goes further in the same amount of time or doesn't go as far in the same amount of time, or the branching looks different. But it was very difficult from those papers to build, at least in my mind, a coherent picture of how the cells actually accomplish ductal elongation and what might be similar or different between how normal epithelial cells remodel the breast versus how malignant epithelial cells spread through the body. And I think one of the big barriers to that, one of the things that made it challenging, was that we had these two really different scales. There was a lot of very careful histology work and analysis of in vivo genetics on the one hand, and on the other hand, there was a lot of work with mammary epithelial cells in culture, because the culture conditions to keep them alive and propagating have been sorted out decades ago. So there's really a lot of good cell biology on the mammary epithelium for a long time. But this issue of 3-dimensional architecture, actually looking at it as a tissue or as an organ, was really very challenging when I started. I didn't begin this field, but it was something that was still quite limited. And I want to highlight that real simply with a couple movies. So if we just take the organ, chop and digest, and we get out the epithelial network, and then digest a bit further, we'll get pieces of epithelium. We call them organoids. And it's important to remember that how they look and how they'll behave is very dependent on the environment into which you put them. So if we take those pieces of epithelial tissue, we put on the bottom of a petri dish, they'll develop something like this, right? They'll land, they'll spread, and then it's kind of a fun movie, right? There's a lot of action. These cells are migrating, they're breaking connections with each other, they're reforming connections with each other, they're dividing. What they are not doing is forming a coherent network of epithelial ducts, right? I don't see anywhere there a tube forming. I joined Zena's lab because I met her at a Gordon conference. She gave a spectacular talk. And what I became convinced was that there was something very important going on in her lab and Immunovisual's lab at the time in terms of trying to model in vivo tissue and organ development with 3-dimensional culture methods. This is about 2002, 2003. So we can do the same thing here. We start with an organ, get the epithelial fraction out, chop and digest down to these organoids, and the only difference, still going to go into the same format culture dish. The only difference is instead of going on the surface of the glass, they're going to be embedded like raisins in jello in a 3-dimensional ECM protein gel. And you'll see the difference is rather dramatic. Instead of hitting the bottom of the dish and spreading out, this piece of epithelium, starting off maybe 100 cells, undergoes a multi-day program of branching morphogenesis. You're watching epithelial ducts initiate, elongate, bifurcate, and polarize in real time. Notice we're imaging this over a period of about 7 days. We can easily make 200 to 300 of these movies at a time with a relatively simple microscope, and we're getting thousands of these per normal mouse. So we're able to directly observe, directly intervene, and try to understand how epithelial cells cooperate and compete to build normal epithelial structures. So what I showed you there is a white light movie. I was doing just a little bit of an advertisement for the power of imaging, right? So that's a white light movie. It's really good at telling you what's happening, right? If I were to add a MEK inhibitor or an ERK inhibitor or add some kind of stromal cells, we'll get a good sense from those white light movies of does the branching occur with a normal speed, normal size, similar number of branches, these sorts of things. It doesn't give you a lot of insight into how the cells are elongating the tissue. For that, it's really helpful to use 3-dimensional confocal or multiphoton microscopy so that you can bring in selective contrast, look at some structures, not others. In this first movie, we're looking at all the nuclei and all the plasma membranes. And what we're able to see is how this group of cells collectively elongates the tissue. It's really nice for some purposes to be able to highlight all the cells, but I'd argue while you see everything, it's also difficult to see any one thing clearly. So we do a number of techniques where we actually intentionally label very small fraction cells, even a single cell, so that we can look at its individual behavior. This is, again, a normal epithelial cell migrating within an epithelial structure as it's elongating one of these buds. And we can even take one step further in and use a specific molecular biosensor to look at the three-dimensional distribution of, in this case, PI3 kinase activity as this epithelial cell is migrating as the duct is elongating. By taking approaches like this, where we can bring in different fluorescent contrast agents, and we can bring in different molecular inhibitors or genetic perturbations, we're able to understand how the cells collectively build the normal organ, right? And what pathways this depends on. Since this is for a pathology department, though, I'm not going to focus on our developmental work. I'm going to focus on our work in cancer. And I'll just use those 3 movies as a launching point for a question that really bothered me for a number of years, which is, why don't these normal migratory cells leave? Actually, go back for just one second, just start that middle movie, right? You look at this cell, it's migrating towards the edge, right? It's clearly able to extend and retract complex protrusions. If you use an actin or a RAS biosensor, you'll see that those are at the tip. If we use electron microscopy, right, we see one of these normal ducts, see a normal epithelial cell reaching out towards the edge, and the only thing keeping it in, from a physical point of view, right, is this couple of little tiny protrusions between adjacent epithelial cells. So I don't think those are strong enough in a mechanical sense to prevent this cell from migrating out. Instead, I think the epithelial cell is detecting a stop signal as it reaches the edge, right? And that's what's maintaining the tissue architecture. If that's true, we should be able to manipulate either the environment or the genetics and get the cell to continue migrating out, leave the epithelium, enter the extracellular matrix. So that's what we're going to focus on doing in the next couple of modules of this talk. The first thing, the simplest thing that I thought would be just this slam dunk experiment. If there's any graduate students in the audience, beware of what the PI thinks is a slam dunk experiment. It's going to work on the first time. Those are often very interesting multi-year projects. So we had this context here where the normal epithelium was forming a multilayer. The cells were adopting this migratory morphology, going right to the edge, right? And then we knew they were stopping. And we knew somehow, some way, metastatic mammary tumors were inducing the cells to leave. Those cells were escaping past the myoepithelium, past the basement membrane, and getting into the distant circulation. So I assumed that if we could just get aggressive tumors, so get cells that were from highly disseminative or even frankly metastatic tumors, we could then put them in these same culture conditions. Sorry about that. Put them in these same culture conditions, and we could directly observe how the cancer cells migrate to distant sites. Right? To do that, we developed a parallel assay. The one I previously showed you was normal mouse mammary epithelium. Here we're using very similar techniques, and we published all of this. If anyone's interested in how we get cells out of organs and grow them, it's in the Nature Protocols paper a couple of years ago. So here we're starting from primary human breast tumors, right? This is always going to be a collaboration between our lab and pathology departments. We both have deep collaborations with the pathology department here. We also work with the CHTN, and I believe have actually received samples from your pathology department through that cooperative network. So we have a 3D organoid assay here. We're going to take what tumor tissue the pathologist will release to us as unnecessary for diagnostic purposes, chop and digest it with very similar approaches, slightly different enzymes, slightly different media conditions, and embed it in a 3D matrix. When we say we're using organoids here, we're doing something kind of different from what Hans Kleber's lab is doing. It's coming from a different tradition. It's coming from west coast of the US, Mina Bissell, xenoworm style approaches. So what we're trying to do here is preserve the in vivo state rather than biobank. We're therefore able to use a simple culture medium because we're not trying to passage anything. And so it's not Wnt-based. There's no strong signal here telling the cells to dedifferentiate. These are acute cultures. They're ready to go in 24 to 48 hours. So you can really go from a sample coming out of a patient to your perturbations, both genetic and pharmacologic, occurring in a matter of a few hours. We get 500 to 5,000 organoids per clinical sample. We've recently started working with GI malignancies, and that number can be much, much higher. 25,000 organoids per PDX tumor, 250,000 per GEMM breast tumor, the genetically engineered mouse model. We have this working in mouse and human primary and metastatic site for breast cancer, and we've since extended to a number of additional malignancies. Okay, with that as preamble, this is a piece of a stage 3C human breast tumor. This particular patient, 20 lymph nodes were sampled, all 20 were positive for cancer cells. So that's as disseminative as I can imagine a patient being without a patient's tumor being, without that patient already having evidence of metastasis at distant sites. However, when I look at this movie, right, I see something closer to branching morphogenesis than protrusive invasion. And I specifically don't see cells leaving into the extracellular matrix here. And I promise, we're good microscopists. We turned the objective turret, looked at higher magnification, looked with specific contrast agents, and we just simply don't see cells leaving in these conditions very often. Occasionally, but not typically. So then I was confused, right? This is a stage 3C breast tumor, extensive dissemination of the regional lymph nodes. Why wouldn't it migrate into the extracellular matrix of this matrix gel assay? And I would like to be able to tell you that I figured that one out overnight. Instead, it took months. And what we were thinking about, what we realized after a lot of thinking was there was nothing wrong with the tissue. It's the goldest of gold standard tissue. There wasn't really anything wrong with the assay. What was wrong was how I was thinking about the assay. I was thinking that if I just got my hands on the right tissue and I put it into reasonably plausible, reasonably realistic 3D environment, that we'd see that type of protrusive invasion we were expecting. And that just wasn't the case. Invasion is not a switch on the wall or a switch inside the genome of the cell that produces a certain set of behaviors irrespective of the environment. Instead, it's a dialogue between features in the tumor microenvironment and properties of the cancer cell. So we thought maybe there was something missing, right, in the 3D invasion assay we were presenting that's typically present around a breast tumor. Since I did my postdoctoral training with Zena Werb, we were very prepared to think about the extracellular matrix, in particular to hypothesize that the strong increase in abundance of collagen 1, specifically fibrillar collagen 1, in the tumor microenvironment might be a signal promoting invasion. To orient you, this is the tumor, and the gold shiny signal, this is this adjacent stroma. The gold shiny signal is fibrillar collagen 1 from a Picker Sirius red stain viewed under cross polarizers. So the birefringent signal is fibrillar collagen. But I need to acknowledge here, when we started this particular study, everyone, nearly everyone in my field of cancer cell biology would have said, okay, if the tumor's not invading, or we see that it is or is not invading, it's probably being regulated by features in the microenvironment here, right? But there would have been, so everyone would have put the arrow here, but the label on the arrow would have been very different from one lab to the next. People would have hypothesized TGF-beta signaling, reactive oxygen, cancer-associated fibroblasts, eosinophils, macrophages, hypoxia, what have you. And so collagen 1 is really only one of several plausible, supported by preclinical studies, hypotheses. So this gives me a chance to highlight an additional advantage of the 3D culture approach, right? It's obvious that it's a terrific platform for visualizing cell behaviors because you're bringing this tissue into close proximity in an objective lens. However, because we're able to generate thousands of replicates per tumor sample, I also want to really leave with you that it's a tremendous way to test different features of the tumor microenvironment for their both absolute and relative impact on cancer cell behavior. So let's take a specific example here, right? Let's hypothesize that the extracellular matrix is determining whether the tumor is able to invade or not. That's a difficult thing to test in a patient, right? It's a very difficult thing to test even in a mouse model because you can't really change collagen 1 for collagen 4 genetically in a mouse without having enormous consequences, not likely a loss of viability, but enormous consequences in every organ. But we can do that very easily in 3D culture. We can take a piece of primary human tumor tissue, chop and digest it, get hundreds to thousands of organoids, and then randomly allocate them to different 3D ECM environments. Treat them with the same media image at the same time so that the only variable here is the 3D extracellular matrix environment. So we're going to compare growing these pieces of human tumor in a basement membrane protein environment, right? It's not exactly a basement membrane, but it's the proteins that would constitute it. Things like laminin 1, laminin 5, collagen 4, versus a stromal ECM environment where it's composed almost exclusively of 3D collagen 1, right? So I'll show you two movies on the next slide. I want you to think of basement membrane versus stromal ECM. If you see any difference between the movies, right, that's due to the composition or it's due to the composition of the extracellular matrix. In the movie on the right, hopefully you're seeing the collagen in the background there. Those little fibers are actually the collagen molecules themselves. they're really quite large. So you're seeing them being manipulated by the cancer cells as they're pulling and tugging and remodeling the matrix and then responding to that remodeling and migrating away. I think this movie on the right here, especially if you keep track of the timer on the bottom right there, is frankly terrifying. It shows you how quickly things can go wrong if you bring the wrong microenvironment together with the wrong cancer cells, right? If they're both actively conspiring to invade, things go quick. I think the movie on the left here is perhaps the most optimistic thing my lab's ever produced, because it's tissue from the same human tumor, and we've repeated this result in breast cancer and several other malignancies. The main outlier is neuroblastoma, which invades much more into matrigel than collagen. But in general, we're seeing a strong increase in invasive behavior in collagen 1. And then on the image on the left here, it's a piece of a human tumor. It has equivalent malignant potential. It would like to be invading, if you will, if you'll forgive me that teleology, but it is constrained because it's in the wrong matrix environment. We can similarly achieve this effect and block invasion by inhibiting FAC or SARC, which are part of how the cell perceives these ECM signals. I want to highlight that it's the current local ECM environment that's important here. It's not a permanent switch, because if we take one of those organoids out of a collagen environment and we return it to we put it in a matrigel environment, surround it with basic membrane proteins, it rounds up and stops invading. So 3D culture is a outstanding platform for studying the tumor microenvironment. We've generalized this approach to other solid cancers such as pancreas, liver, lung, bladder, and neuroblastoma. We're currently using these assays to screen for inhibitors of growth, invasion, dissemination, immune evasion, and distant metastasis formation, using both approved drug libraries and novel compound libraries. And we've generated co-cultures, which I'll show you a bit more on later, to study the effect of blood vessels, fibroblastic stroma, and immune cells. Now let's flip the logic. As important as the tumor microenvironment is, the cancer cells and its genetic changes and its signaling changes are also very important to cancer progression, right? And the tumor is also importantly composed of cancer cells. So what we wanted to do is flip the logic and see if we could do something just to the epithelial cell, right? Normal epithelial cell. What could we do genetically to get it to migrate away past the myopithelium, past the basic membrane, into a suppressive environment, right? In this case, matrigel. So we're stacking the deck against this epithelial cell. There'll be normal myoeps, basic membrane, and a suppressive ECM environment. Can we make single gene changes that are strong enough to overcome that and induce invasion? So we became very interested in the gene Twist1. It is a basic helix transcription factor. It's able to bind DNA, regulate transcription of a variety of genes. It's generally considered a pro-EMT transcription factor. When we started this study, though, it was quite unclear whether it was individually sufficient at the genetic level to induce invasion and dissemination. So what you see here, tissue from the same mouse on the left, without doxycycline, on the right, adding doxycycline to induce expression of Twist1. And you can see there's very rapid induction of invasive behavior. That's the first thing you see. The second thing you see is that not all the cells leave. Third thing you should see, let's watch it again as it loops around, is that some of the cells that leave actually go on to form secondary sites. You see them proliferating and forming these larger structures. This isn't metastasis. Metastasis is a systemic phenotype, requires communication of cells across different compartments in the body, right? And outgrowth in a distant organ. But it's an awful lot of the cell biology of metastasis in a single field of view downstream of a single transcription factor. So we've had a lot of fun using this assay to understand what's changing at the genetic level, at the chromosomal level, right? What are the targets that Twist1 is binding? How is that changing gene expression? How is it changing protein abundance? How is it changing protein signaling in order to convert a stationary epithelial cell to a migratory epithelial cell? It's also turned out to be a fun context in which to try to understand how a invasive luminal cell escapes past the myoepithelium. I thought this would be a particularly appropriate vignette for this audience. So as I'm sure many of you know, the myoepithelium surrounds the luminal epithelium in the breast, and the position of the invasive cancer, of the cancer cells relative to the myoepithelium really determines whether a lesion is viewed as DCIS, so ductal carcinoma in situ, or invasive breast cancer. And we wanted to understand, and it's simply whether the cancer cells are remaining inside the myoepithelium or they've breached the myoepithelial layer and gotten out. So we thought, every pathology department in the world was looking at this relationship between cell types as part of their diagnosis and management of breast patients. We thought it was probably pretty important, and as cell biologists, we should be pretty interested in it. So we realized that we could actually just play a couple of genetic tricks in the mouse and study this in a pretty clean way. Because we knew what the images I showed you of twist1 induced cell dissemination was in a mosaic context where we're driving twist1 in both the luminal population and the myoepithelial population. So everything's disorganized, everything's driving this invasion promoting gene. And you might get a different phenotype, right? It's at least worth hypothesizing that you get a different phenotype if you restricted twist1 expression to the luminal population using this keratin 8 promoter and had a normal myoepithelium present, so that you could just test to what extent is this a defense against invasion. We got a really cool result. And we did Western blots that are in the paper, but I'm not showing that we got similar levels of RTTA expression, similar levels of twist expression, right? But we got a very different phenotypic outcome. If both populations were expressing twist1, we saw almost all organoids disseminated, and they typically disseminated a large number of cells. Conversely, if we restricted twist1 expression to the luminal epithelial compartment only, right? What we found was that very few organoids disseminated, and when they did, it was going to be 1 or 2 cells. So most of those invasive cells were being retained inside the tissue. And that seemed really provocative and exciting, but we couldn't understand how that worked. And so what we decided to do was to play some tricks on the labeling side so that we could have the invasive population labeled in one color, in this case red, and normal myoepithelial cells labeled in a second color, green. I'm going to show you several iterations of this experiment with twist1, with cancer cells, and with E-cad negative highly invasive epithelial cells. But in each case, we have an invasive luminal population, and we have a normal myoepithelial population. So how could this work, right? I'm just going to let this loop twice for you. I'm going to tell you what I think you're seeing here. What I think you're seeing here is a column of red twist1 positive cells getting past the myoepithelium, getting in contact with the extracellular matrix, then being recovered. The myoepithelial cells are migrating to recover those twist1 positive cells and bring them back inside the tissue. I've got a really active 7-year-old son, and he sometimes likes to just sprint off. And I view this as, we're walking down the street, walking down the sidewalk, and he makes a run for the street. You grab his shirt collar, bring him back to safety. So I think what we're seeing here is the myoepithelium is actively constraining the invasion of these twist1 positive cells. But that's a model system. Does it work with cancer cells? Well, in this case, we're using cancer cells from a genetically engineered mouse model. You see they actually got away. They were fully surrounded by the extracellular matrix. One of the myoepithelial cells reached out, grabbed onto it, brings them back inside. And that's a couple cells at a time in both cases. It turns out it also works even when a big front of cells starts to escape and invade into the extracellular matrix. The myoepithelial cells can still reach out, come around it, and bring back tissue integrity. So we think we established 3 important points in this study. First, that we didn't establish the myoepithelium was important. That was really well known. But we established that the myoepithelial cells can actively and efficiently restrain both luminal and basal phenotype cancer invasion using genetically engineered mouse models. That myoepithelial cells can reestablish tissue integrity even after it's been lost. The way I think of this is like a soccer game, right? Rather than a castle wall. If you have a castle wall and you breach that wall, right, the attackers can continue to rush past. But if you kick a ball over the head of the defense on a soccer team, they're not going to give up. They're going to keep trying to bring it past, to bring it back to your side of the field. So it's something where they're actively trying to limit your progress. And this barrier function depends, in data I didn't show, on myoepithelial abundance, intercellular adhesion, and smooth muscle contractility. So it's really a collective cellular defense. All right. So the next thing I want to do is talk about how we can understand which parts of a tumor contribute what, right, to metastasis. I like to say, since I got a very detailed introduction, I like to blame this whole project on my background in physics and my inability to look at a slide like this and understand which part's important, right, and decide which part's important. I can look at it and see lots of things, right? There's dark spindly cells up here in the middle. I've heard people describe them as EMT phenotype cells. There's pale round cells here. I've heard people describe them as cancer stem cells. There's cells invading muscle. There's cells invading fat. You'll see there's lots of pink infiltrates of immune and fibroblastic stroma, right? But I literally remember as a postdoc in training going to a specific mini symposium. There's 5 consecutive speakers, all talking about the same mouse model, MMTV polynomial T, and all telling a different story, essentially They used slightly different images, but essentially about this image saying, this is the key part, this is the key part, this is the key part, right? And they each had their own view on the problem, but there wasn't really an ability to compare and contrast and weigh, relatively speaking, which was most important. And so it was a very hypothesis-driven field at the time, which can be good, but it can also leave you blind to things you didn't think of. What I realized, again, I'd like to say that it was overnight. It took us years of working with the assay to come up with this approach. But what we realized is that we could let the cells teach us which ones are most invasive. We don't need to know anything about it in advance. You can just harvest these organoids. I want you to think of them as really gross ice cream scoops from that tumor, right? So it's just, first organoid might be this area, second organoid, the middle left, third organoid, bottom left, right? And so on across a 2-centimeter tumor, you're getting 250,000 ice cream scoops, right? You put them in a 3-dimensional culture assay and you just ask like, which ones invade? Right? I think it's reasonable to hypothesize that the cells that are actively invading are better at it than the cells that remain in the core. You can think of this by analogy to something like the Boston Marathon, right? If you go to the elite runners division at mile 0, all the runners look fast, right? They all have nice running shoes on. They're all in good shape. They all want to win, right? It's difficult to predict the winner. Conversely, if you go to mile 26.1, you can predict the winner of the Boston Marathon. So it's the guy in the front, right? And he's going to win a 1/10 of a mile later. So that's what we're doing here. We're saying the cell that's invading is probably better at invading than the cell in the middle. This could have been stochastic, right? It could have been that there was no real difference between the cancer cells in one position versus another. And it would have been a really boring line of work, almost a tautology. But it turned out to be pretty interesting. So let me walk you through a couple points. This is a luminal model of, this is a mouse model of luminal breast cancer, right? Oncologists think in terms of luminal and basal breast cancer in echo of the luminal and basal compartments in the normal mammary epithelium and the markers for those cell types. So in a luminal mouse model, it isn't surprising that the cancer cells express luminal epithelial markers, right? It's almost a tautology, right? You've got this, you've called it a luminal model because it expresses genes like K8. What was surprising, in fact, I think very surprising at the time, at least to us, was that the cells that led these invasive columns also expressed basal epithelial genes like keratin-5, keratin-14, P-cadherin, and P63. So they had a luminobasal phenotype. They lacked expression of the full smooth muscle program. So there was no SMA, SMM, calponin. They didn't contract in response to oxytocin. So these are not myoepithelial cells. They're instead a mixed luminobasal epithelial phenotype, similar to that observed in the fetal mammary epithelium. The first thing we wanted to test whether this had anything to do with phenotypes in vivo, right? Was this a weird feature of cancer cells in our assay? And if you look here, blue is going to highlight the luminal cancer cells, red the muscle, and green the basal cancer cells, right? And so the first thing you see is that most of the cancer cells are blue, which tells you that it is fair to call this a luminal model. If you look, however, where they're doing the bad stuff, right, where the cells are actually invading into the adjacent muscle, what you see is that the cells are expressing these basal epithelial markers like keratin-14, very clearly shown in this inset. But check my work here. Look just above that, and you continue to see these invasive columns led by keratin-14 positive cells. The next thing we wanted to do was test whether keratin-14 positive cells led invasion in other mouse models. What you were just looking at was this model of luminal beat, so MT polyimmunity. The GEMM models have this sort of Goldilocks-like quality where there's ones with a little bit of invasion, a medium amount of invasion, and a lot of invasion. And we did this experiment expecting that we'd see different types of different invasive programs in different models. And so it was a true chills down the spine, very rare in my career, chills down the spine moment when the postdoctoral fellow, Kevin Chung, came back with the data and said, we're seeing these keratin-14 positive invasive cells leading columns in these models of HER2 positive luminal B and basal breast cancer. It is not the only way, right, for breast cancer cells to invade. There's alternate molecular strategies that don't rely on keratin-14, but keratin-14-led collective invasion is observed across these mouse models. And as we reported in the paper, it's readily observed across the pathologic subtypes of breast cancer. So you can see it luminal A, luminal B, HER2 overexpressing triple negative breast cancer. So it's a conserved mechanism for cancer invasion. So the first point I want to make summarizing this section is that studying mouse tumor invasion in 3D culture predicted cellular and molecular properties invasion, both in vivo in mice and in patient tissue, right? So that validates the approach we're taking of using these convenient GEMM models to learn things that are deeply conserved about how cancer works that hold true in patients. You always need to check though, because not everything's, some things will be derived features, but a lot of the deep cell biology of intermediate filaments and cell adhesion is really highly conserved. All right, so cells with basal gene expression, such as keratin-14, are observed leading invasion. These keratin-14 positive cells lead invasion in 3D culture and in vivo across mouse models, across breast cancer subtypes. And in the MMTV polyomyxin model, keratin-14 is required both for local invasion and for distant metastasis in vivo. All right, which brings up this big question I got at the end of seminars frequently when I was presenting that work for the first time, which was, how does collective invasion relate to distant metastasis anyway? Right? Invasion is a local feature of the tumor. It's going into the adjacent tissue, adjacent stroma, maybe into the muscle, but it's not directly in the breast. It's not directly going to the bones, the lungs, or the brain, right? So how do you relate one to the other? I've got a whole story here that we're going to tell in 2 slides. And the big shift that I want you to make, you guys aren't that far from Huntsville, this would be a good analogy. So is that this collective invasion column, this group of cells attached to the tumor, right? In a static image, when I just show you those antibody stains, it looks like a static feature, right? But it turns out that's somewhat misleading. It's analogous to a rocket on the launch pad in Cape Canaveral, right? Is a static local feature of the Florida landscape until those engines go off, right? You push the button, the countdown gets 0, you push the button, and then it lifts off, can actually deliver a payload outside our solar system. So what you're seeing here is a group of cancer cells launching from this collective invasion strand, and then potentially at least getting locally, right? We think this is how these nests of cancer cells in the adjacent stroma are generated, at least getting locally, and could in principle travel to distant sites. So we tested that using a lineage analysis strategy that we borrowed from developmental biology. We generated primary tumors with either 2, 4, or 21 different fluorescent colors, right? And then assessed the mixture of colors at the edge of the primary tumor and the circulating tumor cells in early micrometastases as well as macrometastases in the lungs. And what we could say was that in the model we examined, more than 90% of the cancer cells, more than 90% of the metastases are predicted to have arisen from a multicellular seed. So we proposed then a new model for how to think about the metastatic process that we call collective epithelial metastasis, based on collective invasion and dissemination of cancer cell clusters, expression of and requirement for epithelial genes, such as keratin-14, and transitions from keratin-14 negative to keratin-14 positive states to spread through the body, back to keratin-14 negative to drive metastasis. This has some superficial resemblance to the EMT-MET model, but I want to highlight that it's transitions between states of epithelial differentiation, right? We find strong evidence for a hybrid EMT in triple negative breast cancer, data I won't present today, but in the luminal breast cancer, we're typically seeing transitions between epithelial differentiation states. All right. So then we started thinking, what's special about these keratin-14 positive cells? I had never intended in my career to work on intermediate filaments, let alone keratin-14. And so we turned, as we often do, when we have a new problem or having trouble thinking about it, to differential gene expression analysis. And we said, okay, well, let's take these primary tumors. Let's get the keratin-14 positive cancer cells, keratin-14 negative cancer cells, do differential RNA-seq, right, and see what's different. And what we found, opposite of what we were expecting, was that multiple gene families associated with cell-cell and cell matrix adhesion, including those for the desmosome, the hemidesmosome, and the adherence junction, were actually expressed at higher levels in the cells that were traveling through the body. There's multiple different classes we could have started with. I didn't know how to start. I didn't know how to proceed until we understood what E-cadherin was doing. So that's where we began. The conventional model from a bunch of great papers in the 1990s and early 2000s is that E-cadherin is suppressing both invasion and metastasis. This is clearly true in models and in patients for invasive lobular breast cancer and diffuse gastric cancer. It's also clearly a correlation with invasive behavior in culture, right, for cancer cell lines from virtually every organ. The more E-cadherin you see, the less spontaneous invasion. The more invasion you see, the less E-cadherin you expect to find. In fact, in MDA231 cells, they tend to be pretty individualistic and spindly. If you add cadherin, they form clusters, right? When that manuscript came out in the 90s, that was reasonable to expect that it was that move to clusters made them less dangerous. Given work from our lab, Hamer's lab, and Stanger's lab, I think it's reasonable now to think maybe forcing them to be clusters might make them more dangerous. But the bottom line was there was a strong body of work here. And there was a strong body of work coming from pathology that invasive ductal, that E-cadherin is relatively rarely lost, right? Invasive lobular carcinoma loses E-cadherin completely, right? But it is something like 10 to 15% of breast cancer cases, whereas invasive ductal carcinoma is about 85 to 90% of breast cancer. It's typically retaining E-cadherin in the primary tumor and in the distant site. There's some observations of changes in localization from the membrane to intracellular vesicles, but it's typically expressed at high levels. All right, so we're now conflicted. The culture data is saying E-cadherin is an invasion suppressor and suggesting it's a metastasis suppressor. The data from clinical samples is saying E-cadherin is being retained in the metastasis. It means it's hard for it to be a metastasis suppressor at the genetic level. So we wanted to break the tie. We used Rolf Kemmler's E-cadherin allele. We used Liquan Lowe's Cree reporter. And so we built mice where we could readily remove E-cadherin or leave it intact, and decided to just make the comparison, right? If we take away E-cadherin in this model of invasive ductal carcinoma, right, where it would normally be expressed, what happens? On the left, we're leaving E-cadherin intact. On the right, we're taking it away. And you can see those papers were onto something, right? They were right within what they were trying to do. And this is much more invasive. So you take away E-cadherin from this model, you see much more invasion into 3D culture, right? I'll remind you, though, that the version with E-cadherin will metastasize more than 90% of the time in the animal. So while this looks less exciting in 3D culture, it's a pretty dangerous tumor. All right, so we next wanted to test how this worked in vivo. So we're using an orthotopic transplant model, test the requirement in primary invasion and dissemination. And what we see is when we leave E-cadherin intact in this model, actually most of the edge of the tumor is more of a pushing boundary. And it's a relatively small component of it that has this clear infiltrating column sort of appearance. Conversely, when we take away E-cadherin, we convert the edge of the tumor stroma border to being overwhelmingly these infiltrating columns of cells. So we thought that was very consistent with the 3D culture data, at least echoed or rhymed with what people see in invasive lobular carcinoma, and was giving us a pretty clear story, right, that E-cadherin is an invasion suppressor in this model. We next did an in-situ deletion, so we took a tumor that was floxed and had a Cree ER, right, so we could add tamoxifen and induce E-cadherin deletion, allow the tumors to grow to about 10 millimeters, pulsed with tamoxifen, and were able to delete. And what we saw was that on the side of the tumor, right, that happened to not delete E-cadherin, we see these pushing boundaries. The side of the tumor that had deleted E-cadherin, we see these infiltrating columns. So we became convinced that E-cadherin loss was promoting tumor invasion and dissemination, confirming prior literature. We then became very interested in this question of whether it was also promoting metastasis, right? So let's test whether loss of E-cadherin affects metastasis. Same concept, but look at the lungs, right? And what we found was that loss of E-cadherin instead was abrogating metastasis, right? In invasive ductal carcinoma, right, we have significant number of metastases per mouse. When we took away E-cadherin, we see only one mouse with one metastasis. We validated this with orthotopic and tail vein experimental metastasis models in 2 different models of invasive ductal carcinoma. All right, so we observed this uncoupling of the efficiency of invasion and metastasis in E-cadherin deleted cancer cells. There's more invasion, less metastasis. Careful examination of a bunch of intermediates revealed that it was due to a combined effect of deficits in directed cell migration, intravasation, survival and circulation, tumor seeding, and even once normalized for highly inefficient seeding in the distant organ, they were separately also deficient in survival in the distant organ and proliferation in the distant organ. So I want you to think of this like a decathlon where there's 10 events, and these E-cadherin positive cancer cells just do fantastic in the 100-meter dash. They win by seconds, but tear both their hamstrings, right? And they're unable to compete in the rest of the 9 events, and they lose the decathlon. All right, so E-cadherin can function as an invasion suppressor, survival factor, and metastasis promoter in the same tumor. And what we think is going on here is that E-cadherin is actually inhibiting or keeping in check TGF-beta signaling, which would otherwise promote oxidative stress and apoptosis, because when we remove E-cadherin, we get more TGF-beta signaling, more ROS, more apoptosis. Key lessons are that metastasis is a multi-step process. Different cancers could rely upon or need to lose the same gene. Some primary tumors are addicted to epithelial gene expression, and the dissemination through the body is stressful at the molecular level, including at the level of reactive oxygen. And E-cadherin is aiding these cells' survival. All right, the last module I want to talk to you about is a really different feature of these keratin 14 positive cells. It correlates perfectly. I don't know how it's mechanistically linked, but we're going to talk to you about this anyway. So the keratin-14 positive cells have more expression of desmosomal proteins, heavy desmosomal proteins, less expression at the RNA level of the MHC complex. All right? And at the protein level, we see essentially complete mutual exclusion of MHC positivity with keratin-14 positivity. Right? So the keratin-14 negative cells are MHC positive, keratin-14 positive cells are MHC negative. Quadrant 2 is essentially empty. All right. Without MHC class 1 molecules, these cancer cells are going to be essentially, not completely, but essentially invisible to T cells and readily targeted by natural killer cells because the MHC1 acts as an inhibitory receptor to the cytotoxic effects of NK cells. All right, so the first thing we wanted to do was test the effect of natural killer cells in breast cancer invasion. This project was by Isaac Chan, who's a medical oncology fellow in the lab, now runs his own lab at UT Southwestern. So he said, okay, let's put these, let's do monocultures of organoids from the tumor, as well as co-cultures of organoids from a tumor with NK cells from the spleen of a healthy mouse. The first thing I want you to take away here, the difference, these are organoids from the same tumor. On the left, you see vigorous invasion. On the right, all those little dots running around are the NK cells. You see there's a strong limitation of invasive behavior. You'll also see at the very end, it starts invading anyway. And we want to be able to explain both of those things. See a little invasion at the top there. All right, so the apoptotic activity is localizing to the keratin-14 positive invasive strands in the co-culture, right? This is a fluorescence caspase reporter. Over time, they're invading anyway, so we want to be able to explain that. And we wanted to test the effect of natural killer cells also on a model of metastasis formation, as outgrowth from a cluster of cells to a group of cells, right? And what we found was that healthy NK cells dramatically limited the efficiency of colony formation of cancer cells from a couple different models. This also correlated strongly with apoptotic activity. So we're seeing strong increase in caspase cleavage in the presence of the NK cells. And we in fact then next showed that this is really specifically the natural killer cells targeting the keratin-14 positive cancer cells. So now we're not doing a organoid assay, there's no ECM, we're just freshly isolating keratin-14 positive and negative cells and combining them with NK cells in a very short duration assay, just a couple hours, and looking at interferon gamma expression. You see a large increase, right, in the presence of keratin-14 positive cells and background levels of the keratin-14 negative cells. So confirming our hypothesis at the molecular level that the NK cells are targeting the keratin-14 positive cells. Okay, so I told you what was looking like a happy story there, like the breast cancer cells continue to get targeted, right? The most invasive cells are getting targeted by the NK cells. However, right, metastasis is the major thing that's killing breast cancer patients. And breast cancer patients have natural killer cells, at least at some point, right? So how do they ever develop metastases? So this is a great experiment that Isaac came up with, which is let's just take the NK cells from the spleen of a tumor-bearing mouse, right? The same mouse, and see if it has the same effect as NK cells from the spleen of a healthy mouse, right? And it turns out it didn't, right? If you have monoculture, you have a certain amount of colony formation. You add tumor-educated, we're calling them, TENK cells. You see this large increase in colony formation. When we tested this in vivo to see if it was a reasonable effect by tail vein injection and then adoptive transfer of NK cells, we see there's a baseline level of metastasis formation strongly reduced by the presence of healthy NK cells, strongly increased, right, about twofold by the presence of these tumor-educated NK cells. So we wanted to figure out how that worked at the molecular level, did differential gene expression analysis in the K14 negative versus positive, the healthy versus tumor-educated NK cells. And we saw an RNA-seq plot come out unlike any we've ever had in my lab before. We usually get a mixture of like dark blue, light blue, maybe some white across the gene lines. This one's red, blue, blue, red, right? So we're seeing a really complete switch here, made us think there could be an epigenetic change. And when we look, is projected pretty well, not exactly, but pretty well to a difference between active and resting NK cell phenotypes that have been observed in the literature before. All right, so can we stop it, right? That's always the question in cancer. Can we stop this metastasis promoting NK cell phenotype? We took two different approaches there. The first was just what I like to call the thumb down the list approach, and you just look for what's differentially expressed and see if anything interesting pops up. We saw 3 different DNA methyltransferases that gave us a clear hypothesis that inhibiting them could be useful. And then working with Joel Bader and Hildur Knudstøtter in his lab, they were able to map for us all of the predicted receptor ligand interactions between tumor-educated NK cells and either keratin-14 positive or keratin-14 negative cells, which caused us to focus on KLRGI as a candidate NK inhibitory receptor. We use the literature to find TIGIT as a known NK inhibitory receptor. All right, so we're going to use this colony formation assay, going to compare monoculture to co-culture with NK cells. I'm going to walk you through this. It takes just a minute, so bear with me. You've got no co-culture, right? So this is just the cancer cells. That provides the baseline number of colonies. If you add tumor-educated NK cells and an unrelated IgG, you get the enhancement of colony formation from the NK cells. Then we're going to add here in blue whatever the antibody is. We started with anti-PD-1 not because it was well justified, but because everyone was doing it and we knew we'd get asked to. Anti-PD-1 had no effect in this assay. Anti-TIGIT had a nice dose-dependent effect, reducing the colony formation back to baseline, as did anti-KLRGI. But back to baseline isn't good enough, right? We want to really eliminate the tumors in these patients, prevent them from forming. So we next tested whether the combination of DNMT inhibition and either anti-TIGIT or anti-KLRGI could really further reduce colony formation. So again, baseline of monoculture, baseline co-culture can be reduced with TIGIT, reduced further with TIGIT plus decitabine or plus azacitidine. Similar effects with anti-KLRGI plus azacitidine or anti-KLRGI plus azacitidine. So I think what we've shown, data I presented as well as data that's in the paper, that NK cells are early responders to metastatic cells as they arrive in distant organs. NK cells can selectively kill invasive breast cancer cells. Cancer cells can in turn corrupt NK cells to promote metastasis, and that inhibition of TIGIT or KLRGI limits metastatic colony formation. We think this is a potential concept for immunotherapy in breast cancer. A lot more preclinical work is required before it's going to be ready for evaluating patients. All right, at the end here, I just want to give you a couple of quick hits of the types of assays we're building right now that you'll see in papers in a year or two, so you have an idea what we're working on. So we wanted to be able to look at how metastases form in distant organs, and we went back to the future. We borrowed some techniques from literally decades ago in terms of precision cut lung slices. So we're going to load metastases into the lung of the mouse, right? And then harvest the lungs, fill with agarose, cut in a vibrotome, get these sections out, harvest the sections. And we can actually, look at the cell behaviors in a micro versus a macro metastasis as it grows out in the lung parenchyma. The circles are the alveoli. We're currently breeding up the mice with fluorescent biosensors in the lung epithelium and lung stroma, so we're able to do multicolor imaging and really look at how the host cancer cell interactions work. Second thing we're doing is thinking a bunch about blood vessels. So we're working with Peter Searson's lab in material science to build functional blood vessels in these 3D PDMS devices in relationship to functional to groups of primary cancer cells. And what you can see here, we'd actually expected to see clusters of cancer cells detach from the tumor cord, migrate across that space, wiggle between the endothelial cells and migrate away, right? Get taken away by the flow. That was not what we saw. Instead, what we see is that the tumor can, or the group of cancer cells can just reach out a foot, insert in the vessel wall, right? And then persistently disseminate cells into the circulation. We saw analogous structures both in murine and human breast tumors. And the other thing we're doing is we're screening for anti-metastatic drugs. We've got different assays modeling different steps of metastasis. In the screening, we're mostly focusing on the colony formation because we think it's modeling the adjuvant setting, at least to some degree. And we've got it working now where we can robotically plate the assays, robotically treat with different compounds, image automatically, and then run those images through automated scripts to EC50s for the library. It works pretty well. You get images of everything. You can sort it out pretty nicely. And this is an example with that MTV polyo middle T and the approved oncology drug library. You see some drugs have no effect. There's a cascade of different magnitudes of effect and a set of drugs that are very, very effective in blocking colony formation. In the library like that, they're pretty familiar drugs. They're things like Taxol and anthracyclines. But we're doing this now across a series of different models as well as a series of different libraries to try to find more interesting molecules that are highly effective at killing these cancer cells when they're vulnerable, just a few cells before they can form clinically significant metastases. All right, I want to thank the members of my laboratory, postdocs and graduate students. I want to thank you for your attention and thank the funders who've supported this work that allowed it to happen. I look forward to and welcome your questions.
Thank you so much, Dr. Ewald, for joining us today. Many great questions should come from this. Thank you again.
No problem.
Sorry.
Yeah, great, wonderful. A lot of cell biology works. A couple of questions. One, this process, you can almost assume it's highly conserved across different tumor metastases. And you last at the end, you showed some of this DNMTs and all that. I know Steve Bailin and we also worked on an epigenetic modifier called EZH2, which directly inhibits E-cadherin. So I was like excited about that. If you and you know, there are FDA-approved inhibitors for EZH2, any in an angle in that direction to test how that works. Yeah.
Sure. So I think that to the first part, I think that some of the mechanisms we're seeing are definitely conserved across cancer sites. So this requirement for E-cadherin, I think, is going to pop up in a few different papers. The cluster metastasis has definitely been observed in multiple cancers. And the keratin-14 basal gene signature is certainly being used in bladder cancer and certain colorectal cancers, certain gynecologic malignancies, and maybe one type of lung cancer. The paper was a little bit mixed, but I think it supported that idea. I don't want to leave you with the impression that I'm claiming this is how it always works in every cancer type. I think that's been, whenever someone's tried to say my thing is what happens in every cancer, in every patient in the whole world ever, you're certain they're wrong. And so we're not trying to claim that. EZH2 is not something we worked on directly. The connection you're making there is very logical and plausible. I don't have anything to add to it, but I think it would be a very interesting angle to pursue.
We have a question from Maitri. Maitri, you can, I'll go ahead and read it. In the tumor invasion into the vasculature, are these being released as single cells or clusters? Curious how the mechanism is different from the other invasion mechanism you talked about previously.
Sure. So what we've seen in terms of we have two ways of checking what gets released. One is we've got a trap at the end of the blood vessel, essentially like a filter, and we can look just what gets released. And when we look there, we see single cells and clusters. We can't rule out that those single cells might have resulted from clusters breaking apart, but we see both there. What we've actually observed in real time is detachment of clusters. In terms of how does it different from the other invasion mechanism? Do you mean like, is it keratin-14 positive cells that are leading the invasion?
Yes, I was also thinking a little bit about John Condolise's work with the interaction between the endothelial cells and the macrophages.
Absolutely.
So I didn't know, how that might tie into the mechanisms that you might be seeing.
Sure. So we don't have anything specific to comment on yet about how the keratin-14 positive cells relate to this intravasation event. What we just got, the U54 that we just got the end of last year, is specifically to allow us to do these large-scale 3D reconstructions of murine and human tumors and find sites of intravasation, and then use imaging mass cytometry to tell all the different cell types there so that we can precisely locate the phenotype of the leader cell that's invading and all of the various immune cells. I would anticipate from the great work that Dr. Candelas has done that there's going to be macrophages right there. I would also anticipate from great work from other labs that multiple other immune cells are going to also be important.
Thank you.
Lalita?
This is Lalita Suman. My question relates to, and this is very fascinating what you presented, which was on sort of the handoff between Twist and E-cadherin. And it would be great to hear your thoughts on how this plays out in the context of DCIS getting to the point where it transits to being an invasive tumor.
Yeah. So we had a real simple view of this from our own work and from the work of people like Nelly Polyak and Oleg Peterson and Marina Gurkova, that the myoepithelial cells were defending and the invasive cancer cells are trying to get through. There was a paper earlier this year that I imagine some of you guys might have seen. It was in Cell looking at DCIS, and they found that there was somewhat surprising correlation between DCIS structures that had limited myoepithelial coverage and those patients doing well versus ones that had more myoepithelial coverage. They did better, as they did worse. And so I think that one of the things that highlights, because they were studying, of course, the lesion that was removed and then trying to predict whether another part of the epithelium would get a tumor. One of the things that I think that highlights is the importance of the systemic environment, right? I think partly you're going to see from work done at the Mayo Clinic, you're going to see large changes in the myoepithelium and in the structure of the ductal epithelium based on the hormonal environment in the patient, right? You see different patients of the same age might have very different epithelial structures and that'll correlate with different myoepithelium and different types of opportunities, I would say, for invasion. What do we think is going on as those cells invade? I think there's two, I'll tell you directly what we think this week, which is that in luminal breast cancer, it's this keratin, it's typically, right, there's exceptions to every rule in breast cancer, or the breast pathology books wouldn't be so big. But in luminal breast cancer, it's typically going to be a keratin 14 positive basal collective invasion going through those myoepithelial layers. I don't know how the myoepithelium is compromised. I don't know whether they break through, whether they kill the myoepithelium, whether they molecularly co-opt it, or they just outgrow it, right? If you just proliferate more in the luminal cells, you'll eventually outgrow it. We think in triple negative breast cancer, what you're seeing is a hybrid EMT where the cells are vimentin, E-cadherin double positive. K14 seems to matter much less. You're seeing genes like Twist and Slug and Snail and ZEB1 and ZEB2 playing, actually, this manuscript's out for re-review right now, playing distinct roles at distinct stages of metastasis, right? So you're seeing different EMT transcription factors, even from the same subfamily, being required for invasion versus colony formation. So we think there's a sequential series of different EMT programs being activated during metastasis in triple negative breast cancer. Interestingly, that same tumor can have a keratin 14 positive collective epithelial component mixed in, and the clones can be interdigitated in the tumor. So luminal seems simple, collective epithelial led by keratin 14, generally one mechanism running in the tumor. And the triple negative tumors that we've looked at, fresh patient, archival patient, PDX, and GEMM, we're seeing this complexity of multiple different cellular compartments with both a kind of conservative K14 program and this newer hybrid EMT program. And I think that they present very different cellular strategies for invasion from that DCIS to invasive. So longer answer than you're probably expecting, but we think that in those two different contexts, there's really going to be different ways of invading.
One last question, Ahmed.
Yes. Hi, my name is Ahmed. I'm one of the medical oncologists. Incredibly fascinating discussion. Thank you so much for such a detailed input. I guess you really, demonstrated spatial heterogeneity across the tumor growth incredibly well. But what are your thoughts on temporal heterogeneity, essentially clonal plasticity of these cells? Because I think it's also interesting to note that, we know that the events that harbor, the potential ability to, EMT transition and metastatic niche probably happen at a fairly early stage in the cancer. So have that been looked at, at your lab, or yeah?
I would say that you're correctly identifying what we need to do in the next 5 years. This question of cell state, it's a great question. This question of cell state plasticity is twofold. What are the states that a given cancer subtype is moving back and forth between in order to do different steps of metastasis, right? Some of those might be early. We're seeing different EMT programs that might be activated preferentially or be more useful to be activated at a late stage. The second thing is, what are the genetic, or maybe more likely epigenetic, mechanisms that change the plasticity of the cell and makes it more or less able to, if there's 3 states, right? There's some cells that just seem fantastic at transiting back and forth between those states. And there's other cells that seem to get stuck. They can go one step forward and they're kind of, they're stuck in there forever. And they actually make much worse metastasizers. That C3-1 model that was super invasive on the Goldilocks slide, it metastasizes like 5–10% of the time. And we think it's fantastic at getting into this keratin 14 positive program. It actually has a substantial EMT compartment as well. So it's got 2 motors driving towards metastasis, but they're not very good at bulking up distant sites. So the cells can get out, but they don't really, and they look horrific in the primary organ, but they're not good at getting to the distant site and then bulking up at the distant site. So both enumerating what the cell state transitions are and understanding the molecular basis of the the molecular mechanisms that allow you to transit between them. I think those are the 2 biggest things that you're correctly identifying where our work suggests is important, but we haven't figured out yet. So there's still work to do. I don't have to retire yet.
I have to look forward to that, to the work. Thank you.
Thank you very much for joining us today, Dr. Ewald, and thank you, Pathology Department, for being here today.
Yeah, thanks.